Good morning, everyone. Today we're going to talk about titratable acidity, and that's a real pleasure to see that all of you are joining us and are ready to learn about how to measure titratable acidity and making sure you are able to use that tools and the knowledge to use in your own space, in your own winery or in your own environment. Because you will learn about the importance of titratable acidity. And so if you were joining us last time, so about 2 weeks ago, for the first workshop, we talk about pH, so that's related to acidity. And we also talk about the sugar content or total soluble solids. So today we're gonna keep following up, keep talking about that very important topic. So hopefully you're gonna be able to learn more about titratable acidity. And I know that some people don't really consider that much titratable acidity because most of the time we focus on pH and total soluble solid. But you're gonna see how important titratable acidity is in the winemaking process. Also, so we're going to go through some presentation. It's going to last for about 20 minutes, talking about the importance of titratable acidity, but also how to measure titratable acidity and what it means. And after that, we're going to do a demonstration of measurement of titratable acidity using a pH meter. So I think you all received a package that contained the procedure to do that measurement. In addition, with a pipette, with a pipette pump, with the slides of that presentation as well. So hopefully you all have that in hands because you will be able to do the same thing that I'm gonna go through with you. So all the measurement, you will be able to do the same at the same time. At the end of this workshop today, you're going to be able to know how to measure titratable acidity, but especially you will be able to explain how titratable acidity change over time during berry development and ripening. Also, you will be able to describe the optimal titratable acidity values for determining harvest maturity, which is really important when you have a vineyard, but also when you have a winery. Then you will be able to list the aspects of acid composition that have an impact on titratable acidity. You will also learn a little bit about strategies to manage acidity in winemaking, so from juice to finished wine. And then you will be able to know and to understand and recognize the waste management procedures. So what to do when you do lab experiments in terms of waste. And also at the end, we will spend just a little bit of time, so about 10 minutes, to share the values you obtain of titratable acidity and to discuss about those data. So we will go through the calculation of titratable acidity. So what happens during berry development and ripening? And so if you were part of the previous workshop we offered 2 weeks ago, you already have an understanding of that figure. So during berry development, that's when you have a lot of things happening. So you have the berries start to be formed. And so once they are formed, they are really tiny, really hard berries, really green. It's like a rock, but a tiny rock. And then, so during the first stage and the second stage of berry development, that's when you have the size of the berries that increase slightly, but they still are pretty green, pretty hard, until they reach the point of veraison. Veraison, that's a step in the berry development and ripening where everything is gonna change. And so veraison, that's when you have the berries that start to become softer. So you have a lot of chemical reaction that happens, enzymatic reaction that happens, that makes the berries getting softer, but also that makes the berries changing their color. So instead of being green, they start to become a little bit pink and then red for the red grapes. For white grapes, they're gonna start to change to be transformed more into a white, yellowish color. At the same time, at this time point, so veraison, that's when you have a change of chemical composition. that's when you have a decrease of the acidity and an increase of the total soluble solids, also called bricks, bricks. So you have higher concentration of sugar, increase of sugar content, decrease of acidity, but acidity, as we discussed last time, that's related to pH as well. So it's not only titratable acidity, that's also pH. And the pH increase, because the more the pH is, the more neutral, the more basic it's going to be, the less acidic it will be. So that's really all those change. And so as I start to mention, at Verithan, that's when you have a decrease of the titratable acidity. And so just looking at that figure, that's coming from a study that was done on a specific variety in Japan, where they evaluate the change of titratable acidity, but also of tartaric acid and malic acid, because they are the two main organic acids that are found in grapes. And so you can see on the first graph on the top that you have a decrease of the titratable acidity over time, so from Verithan until harvest. You can also see that the tartaric acid concentration decreased, but not that much, compared to the malic acid, which is the bottom graph, where the malic acid is really decreasing over time. So from Verithan to harvest, that's when you have huge decrease of malic acid. And that's really how the titratable acidity decrease over time. Just to give you some numbers and some ideas of values of titratable acidity in different varieties of grapes. So Vitis vinifera grapes, they are the most commonly grown around the world. And they tend to have an acidity, so titratable acidity, that is pretty low in the sense that it will vary between 5 grams per liter to about 9 grams per liter to 10 grams per liter. It depends on where those grapes were grown. It depends on the climate, so it depends on how much sun exposure was there, because the sun has an impact on the decrease or the degradation of malic acid. Compared to Vitis vinifera grape varieties, we put in that table interspecific grape varieties. And so interspecific grape varieties would be the main varieties that are grown in Iowa, in Nebraska, in Minnesota, because most of the varieties are coming from Minnesota. And so just to give you an idea of how titratable acidity is different, you can see that you have a higher titratable acidity in those varieties. And so that range is more between 7 to 15 grams per liter. And that's really because we have much more malic acid in those varieties. They are not degraded that much compared to Vitis vinifera varieties. And that's because of the species or the varieties that are used to create those varieties. They have genes that are related to acidity, and they don't have the same genes as Vitis vinifera varieties. So you can see just by looking at those numbers that interspecific grape varieties tend to be really acidic. And that's quite of a challenge to make wine from those varieties in Iowa and in the Midwest, other states in the Midwest, and even in Colorado. All right, so what is titratable acidity? That's the main question. Why do we care about titratable acidity? And so titratable acidity, that's what is defined as the number of protons. And that's a little bit in depth in the chemistry, but that's really important. So that's defined as the number of protons that are recovered during a titration with a strong base. And so if you already started to do some analysis of titratable acidity, you know probably that we use sodium hydroxide, which is a base. And so by using sodium hydroxide, the structure of sodium hydroxide is able to titrate and to act on the protons of tartaric acid, which is the main organic acid, and malic acid. And so we do a titration to evaluate how much protons do we have. That's how we define titratable acidity. And that's really related to the concentration of acids we have, which is different as the pH, because the pH gives you an idea of the acidity, but it doesn't give you an indication of the strength of the acidity, how much acids do you have? And so the pH, that's really the measure of hydrogen ion activity, which is different. And two solutions with the same titratable acidity can have a different pH. And that's really because it doesn't look exactly at the same thing. Something to note is, and that's something I'm going to mention in the next slide, is the pKa. So pKa, that's the pH at which a weak acid is half dissociated. And that's what we do when we do a titration. We look at the pKa, so how it's changing the pKa during a titration. And so the lowest the pKa, the strongest the acid is. And the highest the pKa, the weakest the acid is. And so that's how it's related to the strength of the acidity, the concentration of acids. So organic acids in grapes and wines, you know, I think, the main one, because I mentioned that, that's tartaric acid. So tartaric acid, that's the main organic acid that is found in grapes. That's how the oldest wine has been discovered. It is 8000 BC that was found in the Middle East. Explorers discovered that old wine because when they looked at a jar, inside the jar, they found some crystals. And when they did analyze the crystals, it was the potassium bitartrate crystals. And so that was containing tartaric acid. And because tartaric acid is the acid in grapes, that's how they were able to identify the oldest wine. That was just the little story. But tartaric acid is the main organic acid. And so it has two different pKs, because if you look at the chemical structure, you can see you have one proton and a second proton. That's really what provides the two pK. That's really what is going to help us identifying how much titratable acidity is if we only have a solution of tartaric acid. You can see the pKa is pretty low compared to the pKa of malic acid. And as I said earlier, the lowest the pKa, the strongest the acidity. So tartaric acid is naturally present in grapes. This is the strongest acid in grapes. Then you have malic acid. That is a little bit weaker, so less strong as tartaric acid, but that's the second main organic acid found in grapes. Then you can have a little bit of citric acid, and that's what you can find at a concentration that is much lower than the two others. You can have less than 1 gram per liter compared to tartaric acid, which can be about 10 grams per liter. Same for malic acid, you can have up to 20, depending on the berry ripening stage, 20 grams per liter of malic acid in grapes. Another organic acid you have in wine, that's succinic acid, and that's an acid that is formed during fermentation. Then you can have lactic acid, which is formed by bacteria. So when you do malolactic fermentation, which is a second type of fermentation, to reduce the acidity of wines, that's going to form lactic acid. And that's really because of lactic acid bacteria that we use to do that. And another organic acid you can have in wine is acetic acid. And so you probably know that acetic acid is really related to the vinegar smell. That's the main acid in vinegar. And so that's being produced by bacteria. And that's an indication of spoilage. If you have a too high concentration, that's going to lead to a flaw and sometimes a fault in wine. And so all those acids, they are all part of the titratable acidity. And so that's really what you evaluate when you do titratable acidity analysis. So why is it important to look at titratable acidity? That's because, as I explained, titratable acidity has a strong correlation with the sourness, the acidic perception, the strong acidity, the concentration of acids. So the more titratable acidity you have, the highest you have, the more acidic your wine is going to be perceived. That's really important to determine when it's time to harvest your grapes and the style of wine. So if you have a grape that has a titratable acidity that is at 20 grams per liter, you may decide to wait before harvesting your grapes. That's also important just to know and to identify what type of treatment, most adjustment you might need to do to reduce a little bit this acidity. And so that's one of the points that you may identify. If it's a high titratable acidity, you need to de-acidify your juice or your wine. And so there are a lot of different techniques to do that. You can add potassium bicarbonate in juice to reduce that acidity, so to de-acidify. You may do malolactic fermentation to change the organic acid that is found and to change that acidic sourness perception. You may do blending. You may do amelioration, which is adding water to do a dilution, basically, of the acidity. So there are many different techniques to de-acidify or to acidify. If your titratable acidity is too low, you may need to add some acids, organic acids. Also, when you realize that probably you need to acidify your wines, there are some rules that you have to follow based on the Tax and Trade Bureau of Alcohol and Tobacco, so TTB. So there are a minimum of acids you can add and maximum of acids you can have in a wine. And so the maximum would be 9 to 11 grams per liter of adding acids. So make sure you know those rules before making any adjustment. So how can we measure acid content? And so the first type of measurement, as I explained, that's titratable acidity. And so that can be done by using a pH meter and an electrode, and that's what we're going to do today. If you don't have a pH meter, you might be able to measure titratable acidity using an indicator, a color indicator, which is phenolphthalein. And so that's really just to observe a change of pH by looking at the change of color. So that's really, if you don't have a pH meter, you might be able to use that indicator. However, if you are measuring titratable acidity in a red wine, the change of color might be a little bit trickier to observe if you use the color indicator. So just keep that in mind. Another way to measure acids and acid content, that's using either enzymatic measurement. So looking at how the enzymatic activity is based on the different types of organic acid you have. Or you can use a really much more expensive equipment that we have in labs, but you most likely don't have. That's high performance liquid chromatography. And that's really just to have a better idea of what organic acids do we have in juice and wine, and in which concentration do we have those. So that's a pretty specific equipment and specific method. So before we move on the demonstration, I want to make sure we all know the safety rules. And so we're going to go through those safety and best practices, especially so for us, we are in a teaching lab in the Department of Food Science and Human Nutrition at Iowa State University. And so we have to wear lab coats, we have to wear gloves, we have to make sure all the protective personal equipment is handled correctly. So probably you don't have a lab coat. If you do, feel free to use a lab coat. If you have gloves, that's one of the things that's really important to wear, especially when you do titratable acidity measurement. That's because you're going to use sodium hydroxide, which is a base, as I explained. And so base is really corrosive, same as acids. And so if you don't wear any protection, any PPE, you might have some sodium hydroxide in your hands. And then if you touch your eyes, you're going to have sodium hydroxide in your eyes, which is really damaging. And that can be pretty bad at some point. So feel free to wear gloves if you have gloves. Make sure you have closed-toe shoes and long pants, making sure all your skins is protected. Also, so when you start to be ready and you try to be ready to do any measurement, when you use a pH meter, make sure, and burette and all those equipment, glassware, make sure you rinse all your equipment with deionized water, just to clean everything, to rinse any particles that could stay there and that could be negative at some point for your future measurements. When you use a burette, and we're gonna talk a little bit more about that during the demonstration, but when you use a burette, that's where you add your sodium hydroxide, where you add your base. And so make sure when you do that, there is a tap at the bottom. So you need to make sure the tap is closed before filling the burette, but also the tip of the burette should not have any bubbles because that might change the measurement. When you're gonna be looking at how much volume you have in your burette, it's gonna form something round. And so you would have to look at the volume, looking at the bottom of the meniscus, that's what it's called. So at the bottom of that type of bubbles. So make sure you read always at the bottom, and to do so in a appropriate way, you should make sure your eyes are right in front of that line. And so what we're gonna do today is measuring titratable acidity in the grape juice we did provide you. And so you will be asked to record all the different values. So that's always something we do in grape and wine chemistry or in a lab, in a winery lab. That's keeping track of everything, keeping track of all your data, the date of your analysis, what samples did you have, like which grape did you collect, how did you collect those grapes, and then did you crush a pool of grapes to evaluate if it's time to harvest, and where was it coming from. And so that's exactly the same thing we're going to do today, making sure you keep the data. You record all your data. So you're going to be asked to, on a piece of paper, make sure you're going to be able to record the initial volume of your burette, the final volume of your burette, and then you will calculate the total volume that is used. You will be able to write the normality, so which is the strength of sodium hydroxide we're going to use. And also the sample, the volume of your sample, so the volume of your grape juice. And then on another column, you will be able to calculate the titratable acidity. So be ready with a piece of paper. Somebody is going to take notes if you are two, or if you are alone, you're going to have to take notes. Once we are going to be done with the experiment, with the analysis, there is a lot of things to do with the waste, because we're going to get a little bit of waste from sodium hydroxide, but also the juice, grape juice. And also, if you have a pH meter, depending on the type of pH meter, you might have a solution of storage. And so this solution of storage contains some silver chloride, which is pretty nefast or negative for the aquatic life. And so that's something you don't want to throw in the sink. So I'm not sure how you handle the waste from labs, but make sure you handle it as it's appropriate. And so to learn more about waste management, you can go through the safety data sheet that are coming with the sodium hydroxide solution. So with all the chemicals you purchase, you should have those safety data sheets. So make sure you have a good understanding of how to manage that waste. We're gonna start talking about the demonstration. So we have first a burette. And so I'm just gonna show you the burette. So here I have a burette of 50 milliliter. But the burette, that's really that cylinder that has some graduation on it with a tap and with the tip. All right, so if you have that, make sure you have the burette on a stand, because that burette should stay always straight. If it's not straight, you're not going to be able to have enough sodium hydroxide or not appropriate sodium hydroxide concentration solution. You should also have a beaker. So a beaker, that's a glassware. If you don't have a beaker, but if you have a same type of thing with a flat area, that's what you're going to use to do the titration. If you have a stir bar and a stir plate, and so that's to make sure while we do the titration, everything is being shaked. Because if we don't shake, over time, the titration won't be homogeneous. And so the measurement won't be accurate. So you should also have received a pump, pipette pump. So make sure you take that pipette pump. So it has different pieces, but there is a wheel that is going to be used to pump the juice. And you should also have received a pipette of 5 milliliters. So you're going to put that pipette on top of the pipette pump. So make sure it's handled correctly and it's closed and easy to hold. So that's what you're going to use to pipette the juice. So that's what you should have. You should have a pH meter. If you don't have a pH meter, make sure you have the phenolphthalein, the color indicator I mentioned earlier. Then, if you have a pH meter, so you should have buffer solution of pH meter, because that's going to be the first step that we do, is calibrating the pH meter. So here in our lab, we have 2 different pH buffer solution. So we have pH 4.0. That's a standardized solution buffer. And we have pH 7. And that's really the first step. When we do any measurement with pH meter, we always start by a calibration of the pH meter. So if you have those buffer, make sure you transfer into a smaller beaker or in a tube. And make sure you pour enough in your beaker or in the tube to be able to have the entire probe. So the electrode should be in contact with the entire solution. So make sure you have enough volume. And that's probably 30 milliliter that you may need in those beakers. The good thing about the buffer solution is that they have different color. So you can see the pH 7 is yellow and the pH 4 is pink. If you forget to mention anything on your beaker, you will still be able to see the difference. So you will be able to know which solution you're going to use. So to start doing calibration, you might need to remove the storage solution. So in our pH, at the end of the probe of the electrode, we have a container that has a storage solution. So make sure you remove that and you put on the side. So that's the electrode that we have. Make sure you move that part away because you will need to have a big piece of the electrode in contact with the juice or in contact with the buffers. Also, you might have a little hole on your electrode. If you do have a hole, make sure it's open. And that's really just to make sure the pressure is going to be the same between the inside of the electrode and the outside. So make sure that's open. So you should also have a water bottle, so deionized water. So we have those fancy bottles, but probably if you don't have this one, that is totally fine, but make sure you have a way to use some water to rinse, because that's what we are going to do first, is rinsing the electrode with GI water. And so to rinse, you need to have a waste beaker to gather all the liquid. All right, so I'm going to rinse first. Make sure you don't put water anywhere else except for rinsing. Once you rinse, after you can dry. And to dry that, to make sure you don't have any remaining water, you can use a soft tissue or a clean wipe, so really thin, to avoid scratching anything. So you're gonna make sure this is dry. And so on your pH meter, you should have a way to see, I can try to show you. So that's the pH meter. You should have a lot of different buttons. One of them would be either standardized, so STD, or Calib, or Cal, C-A-L. And so that's the first things we're going to do, is pushing that button, either Cal or standardized, to start the calibration. And most of the pH meter are going to mention which buffer solution you should use first, because sometimes you should use the pH 4 first. Sometimes you should use the pH 7. To calibrate, you should make sure the entire probe is within the solution, and also make sure you have a steering movement. So if you don't have a steer plate and a steer bar, you can steer just by steering. So make sure it's always in movement. Make sure it's always shaking. And so you keep an eye on your pH meter until the value is stable. The value should be stable at 4.01 if you are using the buffer of 4.01. Once it's stable, you're gonna press again the Calib or the Cal or the standardize or the accept button. So it really varies depending on the pH meter. One thing to consider as well is the temperature. Some probes are gonna have already a temperature probe. Some other probe will have a temperature probe on the side. So you will have to make sure your solution is always in the same, under the same temperature. So it's always with the temperature being measured. So steer fast or slow. I would say steer, that's hard to define, but fast, if you go too fast, you can bring too much oxygen or too much air, bubbles, that will change a little bit the value of the pH. So make sure you still steer in a way where you make sure everything is in contact with the probe, but without bringing too much bubbles. So once you use the first pH and you did the calibration on the first pH solution, then you rinse again with your DI water. You clean or you dry again. And that's really before moving on the next one, the next buffer solution. So I'm done with pH 4.01, so I'm switching to pH 7.0. And it's gonna stabilize or calibrate again. So I keep steering. I keep making sure the probe is within the solution. All right, and it is stable for me, so I do the calibration. So for those of you who did the calibration, so make sure you, I think I did rinse, but make sure once again, once it's done, you accept the calibration. And so you should have a value in between 95% and 100%. And that's really just to give you an indication of the calibration. If that's a correct calibration, you should be close to 100%. If that's not a good calibration, you might have less than 90%, which means you would have to redo things. Just making sure the calibration is done correctly, because that's going to really impact the final values, the final titratable acidity. So now that the pH meter has been calibrated, we're going to start preparing for titratable acidity. So what we need is first to fill in the burette with sodium hydroxide. And so if you use pellets, you might need to create or to develop that sodium hydroxide solution, because we need a liquid. And so to do so, you will have to make sure that's standardized. So the strength of sodium hydroxide is correct. And so there is a procedure to standardize sodium hydroxide in the handout we shared with you, we sent you. So make sure you go through that step. So sodium hydroxide, so I'm going to use, so that's coming from a standardized solution, but that's just for ease of use. So we are going to use a 0.1 normal. Normal, so N, is the strength of sodium hydroxide. So that's the solution we're going to use today. Before filling in the burette with our solution, we need to make sure the tap is closed, because we don't want to have everything leaking everywhere. We want to make sure we have a waste container, like a beaker at the bottom, once again, just to make sure nothing is leaking everywhere, especially because it's going to be a base, corrosive, and so you don't want to have that solution anywhere in your working space. And then here I'm using, as I said, a 50 milliliter burette, which is pretty big, pretty tall, right? And so to avoid spilling solution everywhere, you might need to use a funnel, unless you are really like a pro to do that. I'm not really a pro, so I'm going to use a funnel. And so I'm going to just transfer almost all the solution. Once again, make sure the tap is closed. And so I won't put everything, because I don't need everything today. If you know that you have a lot of different analyses to do, you can fill in the entire burette. That way you won't have to refill that many times. But for today, because that's only one measurement, we're just gonna use a little bit of it. And so I have a specific level, and so that's where we will be able to look at, and that's what really I was So that's what I was explaining earlier. To evaluate the volume you have in your burette, you might have to look through, like at the same level. Your eye should be at the same level as the bottom of that shape. For accuracy, you look in the middle, at the bottom. So we have our solution of sodium hydroxide. The first step we're going to do is to make sure the water we're going to use is going to be at a correct pH. So it's going to be standardized as well. So in a beaker, the beaker you're going to use to do the titration, you will need to have a stir bar if you have a stir plate. If you don't have a stir plate, you're gonna have to do it by yourself. It can be challenging because you will have to do 3 things at the same time. So it's probably worthy in investing into a stir bar and a stir plate. You have a lot of different size of stir plate. It doesn't have to be big. So on the stir plate, you put the beaker and the stir bar. In that beaker, you're gonna use 100 milliliter of DI water. And so to do so, you can use a graduated cylinder. Here, that's a graduated cylinder of 50 milliliter max. So I'm gonna do twice to get 100. And so I'm gonna pour. And to make sure I have the correct volume, I can use a transfer pipette to evaluate how much I should remove or how much I should add. And so with the pipette, I'm gonna put myself in front, my eyes are gonna be in front of that meniscus, and I still need to add more. So I'm gonna use the pipette to make sure I reach up to the line of 50 or 100, if you have 100. All right, I've got 100 or 50, sorry. I'm doing it twice, so I'm just transferring, making sure I have 100 milliliter of water in that beaker. All right. I've got 100. So that's what we're going to start by doing, is looking at the pH, making sure the pH of that solution, we reach out to 8.2. pH 8.2, that's the target, by adding sodium hydroxide. But to do so, we need to make sure the solution is under stirring, and also that we put the pH probe. It's stirring very fast, so I'm stopping for the moment. But the pH probe and temperature probe, if you have 2 separate things. So making sure the probe is in the solution, but doesn't touch the stir bar. Because if you start stirring, it can break. If the stir bar touches the probe, but that's a piece of glass at the bottom of the probe. And so if you have the stir bar touching, that might break. So make sure the probe is within the solution and the stir bar is not touching it. In addition to that, make sure you have the tip of the burette on top, so not touching the wall of the beaker, but just on top of the solution. So that's 3 different things, 3 setups. Once you have that, look at the pH you have. So click measure on the pH meter and see if you have a pH that is stable first. And that is 8.2. That's the goal. If you don't have pH 8.2, you will add a little bit of sodium hydroxide. So with the tap, you're going to open slightly to make sure you have at least 1 drop that goes into your solution to get the pH of 8.2. And that's really just to standardize. And so once again, if you stir fast, it's going to bring some air. And so that might be providing an inaccurate reading. All right, so once you have the pH that is correct, you're going to add the juice, because that's what we want. We want to evaluate how much titratable acidity do we have in the juice. So to do so, we take the juice, so we just transfer the juice we provided to you in a tube. for ease of handling that. And so you're going to use the pipette that you put with the pipette pump. The goal is to get 5 milliliter of juice. So 5 milliliter, that means in this pipette, you will have to get 0, which is, that sounds weird, but that's how it's measured. So 5 milliliter. And so to take that, you will use the little pump. So you will use that wheel to move down to have the juice getting into the pipette. So you probably see that I'm pipetting the juice up to 5 or 0. Once I have that, I will just transfer directly in the beaker, and I can push here to release the juice. All right. You might still have a little drop in the pipette, but that's part of the error of the pipette. So don't touch that little drop. You don't need that drop. Once you're done, you can remove the pump, because if you keep having the pump with the pipette and you move, like you turn that way, that's where you're gonna get juice into your pipette. Juice is really sugary, really sweet, but that can lead to a stuck pump at some point. So make sure you don't have juice getting into the pump. Always use that pump straight. All right, so you can put that on the side. So in your beaker, you should have the stir bar, if you do have, the water, 100 milliliter of water, the 5 milliliter of juice, and record 5 milliliter on your notes. Take notes of the volume of juice you used. Then the first thing you need to do is looking at what is your volume in your burette. And that's what we call the initial volume. And so here I have, so making sure the graduation is easy for you to look at. And I look really at the same level. My eyes are right in front of the line. And so I'm looking at the bottom of the meniscus, and I have 24.1. Maybe make sure that you read in the correct way the graduation. That's a source of error. Sometimes you would have the graduation, the numbers are going to go up when you go up on the burette. Sometimes it will go down, in the sense that here I'm 24 and then 25, 26, 27. Sometimes you will have 24, 25, 26, 27. So make sure when you do the reading of the initial volume, you read correctly. So I'm going to record 24.1. That's my initial volume. Make sure you record your initial volume. And so now we're going to start. So make sure you stir your solution and you look at the pH. The goal now is to start the titration. I'm stopping because it goes so fast, but the goal is to do the titration. So it's to add sodium hydroxide slowly until we reach the pH of 8.2. That can go fast. As soon as you reach pH 6, that might be very fast. In just one drop, you might have pH 8.2. So make sure you keep an eye on the pH meter all the time. And so you can, so make sure it's under stirring and open the tap, but gently, to make sure it goes drop by drop. Don't go too fast with the tap. If you have like a a splash of sodium hydroxide, you might be way over the pH of 8.2 too fast. So make sure you keep an eye on the pH. As I said, once it gets close to pH 6, in few drops, you're going to have a really high increase of the pH. So always do it under stirring, and that's where it's beneficial to have a stir bar, because if you have to check the pH, do the titration, so opening the tap and stir at the same time, that won't be really easy to do. So you keep going until you have, the goal is pH 8.2, 8.20. If you have 8.25, that is fine. If you have 8.18, that is fine too. But don't be at pH 9, because that's going to be inaccurate. So to me, I'm done. I'm at pH 8. I'm a little bit below, above, sorry. But I consider that I'm good. And once I'm done with the pH, when I reach the correct pH, I will record the final volume. And so I will go back on my burette and check once again, what is the volume of sodium hydroxide in the burette that I have. So look again at the meniscus and record that value, which is the final volume. So I've got 25.2. So if at some point you go over the pH of 8.2, you will have to redo the experiment. And I would say anyway, it's better to do at least 2 times that measurement, because you can have an error the first time you do it. So make sure you do 2 or 3 times the same analysis on the same juice. That way you can do an average after and really identifying the representative value rather than just one measurement. Also, when you do titratable acidity analysis in grape juice to evaluate when it's time to harvest, it's much better to have a large representative grape sample, in the sense that you should go in your vineyard and gather grapes from different locations within the same row, from different vines, from different rows. So make sure you get a pool, a real representation of your samples. And then you make juice from that pool of different clusters, rather than getting juice from just one cluster. So I've got a 50 milliliter burette. That is correct. 50 milliliter burette. You can have a 25 milliliter burette. That is fine. The volume really doesn't involve anything, doesn't induce anything. Just if you plan to do a lot of different analysis, that's good to have a big burette, a larger volume, because you can do multiple analysis with the same solution of sodium hydroxide. So once you're done, make sure you keep something under the burette, because you still have sodium hydroxide in that solution, in that burette. So make sure if it drips, you have a waste container under, or you still have your beaker under. Then you need to clean the pH probe all the time. You need to clean before putting back to storage. So same as before, you have your waste container, you have your DI water, and you rinse your pH probe. After rinsing, you make sure you dry it. So with a Kim wipe or a soft tissue or soft paper towel. And then you don't forget to close the hole, because if you forget, the inside, the liquid, if you have liquid inside or gel, it's gonna dry. So make sure you close each time when you're done, when it goes to storage. And then you can put that back down. And you can put the storage solution back. Make sure your container is full of storage solution and it's not moldy. Sometimes you might see black spots. That means it's moldy, and that means it's not good anymore. So you will need to clean, to empty it, to clean, and then to refill. And so make sure the entire probe is in this solution, in contact with the solution, because you don't want to let it dry. Because if your probe gets dry, it won't work very well anymore. All right, and so then you're done. You can turn off your pH meter. So based on waste management discussion we explained, make sure you know what to do with your waste. So you can use a waste container that is gonna probably be picked up by someone, and that I'm not sure how you do in a winery or in your own space. And then, so you will need to make sure the burette is empty. And so you will empty. So I'm just taking the initial container of sodium hydroxide. You can empty in the trash, or you can keep reusing it. But make sure you don't wait too long between two uses, because at some point some water is gonna be in contact with the sodium hydroxide solution, and the strength is gonna be different. So make sure you follow the recommendation from the provider. And then, so one other thing is, once the burette is empty from sodium hydroxide, make sure you clean all the burette with DI water. Once again, you don't want to have crystals being formed. So make sure you rinse. You use the burette and you rinse. And I should have kept my gloves, but. And so you keep having a beaker under and you clean with water. And for me, that's easy because I have that fancy burette, fancy pipette. But make sure you do multiple rounds of cleaning. So the final reading for me, it was 25.2 milliliter. That was my value, but it doesn't mean that's the value you're gonna have, because it depends on the strength of sodium hydroxide you have. It depends on many different factors. And so to calculate the titratable acidity value, you need to use the volume of sodium hydroxide that we used. And so in my case, it was 1.1 milliliter of sodium hydroxide. So to come up with that volume, you use the final volume that you subtract with the initial volume. And that's going to give you the volume of sodium hydroxide that was used. Then you multiply that value by the normality of sodium hydroxide, so by the N value. In my case, it was 0.1 normal. Some people might use 1 normal, which is really strong, and so the titration might not be that accurate because you're going to have a huge change of pH in just 1 drop. So if you have a 1 normal sodium hydroxide solution, make sure you dilute 10 times to make sure you have the 0.1 normal. But if you use 1 normal, you can just use 1 normal. And then you're going to multiply by 75. And so 75, that's a factor that is considering the number of protons that we have in tartaric acid and in organic acids overall, and the number of protons that is necessary from sodium hydroxide. So that's a factor that is used when we want to express titratable acidity as tartaric acid equivalent. If we were going to express as malic acid, that would be a different value, because the molecular weight or the weight of molecules of malic acid is different than of tartaric acid. So make sure you know which factor you use. And then you're going to divide all of that by the volume of juice you use or wine you use. And so in our case, it was 5 milliliter. So to me, it was 1.65 grams per liter. Who else wants to share? You can unmute yourself if you want, just to say your number. And I think I'm kind of happy to see that the values are different for all of us, because that gives us a little bit more discussion and more explanation about the errors that we might have. But overall, you can see that we have anything between 0.5 to 2.4. And so 2.4, as it was explained, that was using the phenolphthalein. And so the values was probably a little bit less accurate because it was not looking exactly at the same pH. It was not looking at a value, the strict value. Otherwise, 3 values are in between 1.65 and 1.7, so 1.6, 1.7. So that's how it's really important to do duplicate or triplicate the analysis just to see how is the variation. But that big variation can come from either a bubble that was in the tip of the burette or a reading that wasn't done exactly the same way in the bottom of the meniscus. That can be also, maybe you had some air when it was steering. If you were using a steer bar that was going really fast, you might get some air. And so the reading of the pH might have not been the best. Also, a big issue could be, or a big challenge is to make sure you go drop by drop. So when you do the titration, really go slowly, because if you just add 2 drops or even more, the pH is going to change dramatically. And so the final value won't be accurate, won't be correct. Other sources of error could be in the pipetting of the juice. So if the goal is to use 5 milliliter of juice, but depending on how you pipette, if you are taking a little bit less or a little bit more, you're going to induce an error in the calculation. And so your final titratable acidity might not be the best. You can also have an issue with the pH meter calibration. And that was one comment I think James made about the pH meter not being calibrating correctly. And so that's a source of error. If you don't calibrate your pH meter before doing any analysis, you won't have a correct value. So that's really the most important step, I would say, in the titration. That's really making sure the equipment you're using is calibrated correctly. If you have issues to calibrate, make sure you go back on the recommendation or on the procedure for your specific pH meter. Just making sure that's calibrated the way it's supposed to be for your pH meter, because they are all different. And so that's difficult sometimes just to explain what to do, because you have different buttons to push and all of that. But also another challenge could be the temperature. So most of the pH probe are going to contain already a temperature probe. And so the pH is related to temperature and to pressure. That's the physics aspect of things. But if you have not a temperature probe included, you might have to use a temperature probe on the side, and making sure you do the adjustment of temperature. So that's another potential issue. Before closing, I would like to thank you all for participating today. I hope it was a great way for you to learn, and I think it was a great way for you to learn, because the goal that's really to provide you all the knowledge, but without having to drive 2-3 hours, without spending a full day with us. So that's really the reason behind offering those virtual live workshops. So also thank you to Iowa State University Extension and Outreach, who was sponsoring that workshop development. It was a great pleasure to show you all of titratable acidity. If you have any other questions coming up, feel free to email us. We are going to respond to your email with those information. Thanks again, everyone, and have a good day and see you next week. Bye.